Hello everybody, my name is Ville Hoikkala. I'm the head of Bioinformatics and a co-founder in a Finnish phage company called PrecisionPhage. And my topic for today is about genomic monitoring of phages. And I've mentioned GMP-compatible in this title. I've chosen it because GMP is the most strict framework in this phage field, but what I'm about to present to you also would apply to magistral production or any GMP-like framework as well. Full disclosure, we do have as a company a financial interest in this topic, which I will be presenting an upcoming product that we have in the pipeline towards the end of the presentation. But I would like to keep this as generalized as possible and invite people to discuss these matters as a community as well. So I'm going to start off with a brief introduction because I don't think many of you know who I am. So I have an academic background. I've been involved with phages for roughly 12 years now. I started with doing computational modeling of phage therapy as my master's, and then I did my PhD on phage bacterium coevolution, looking at CRISPR-Cas systems in Lottarina Sundberg's group in the University of Jyväskylä. I then did my first postdoc with Matti Alasvuori in the same university doing reproducible phage bioinformatics. And then I did my second postdoc in Malcolm White's group in Scotland. And I was also working on phage bioinformatic tools. During my first postdoc, I participated in co-founding PrecisionPhage. It was co-founded in 2022 as a spin-off from the University of Jyväskylä. We provide bioinformatic tools and services, as well as phage licensing and phage lab services as well. As I said, I'm the head of bioinformatics, so this talk will focus on bioinformatics. So, phages and genomes. What is a genome? Why is it relevant? And why should we sequence it? To make sure we're on the same page, I'm going to start with the very basics. So a genome is made of nucleotides, typically A, T, G, and C. And certain stretches of these nucleotides are classified as genes, giving rise to proteins, which can be considered the building blocks of life. And other stretches of a genome may serve other functions, like regulatory functions between the genes. Now I'm going to put forward this bold philosophical statement saying that the genome is the phage. So if you think about a lytic phage infection, as the phage injects its DNA or RNA inside the host cell, the phage virion, the capsid, is discarded, and the phage continues its life as a genome inside the bacterium until it's replicated and packaged into new virions, which then spread to the environment. So the genome is basically the only permanent component, should I say, of the phage. So it's a very central thing. Phage genome sequencing has a very long history. The first genome ever to be sequenced was a phage in the 1970s, an RNA phage. And as with any genomics, new methods are making phage genome sequencing very cheap and very fast. And since these are really small genomes, it doesn't really cost a lot of money to sequence a phage these days. Now, as the genome is a very central part of a phage, it also has some implications on the phage production as well. So I'll be talking about phage production now and the emerging standards in phage production. If you think about bacterial production, you start with a research cell bank, and here I'm using GMP as a framework. You start with a research cell bank, and when you enter the realm of GMP, you create a master cell bank from this research cell bank. And from the master cell bank, you then derive working cell banks from, and from these working cell banks, you would then create your end product. With phages, the terminology in such a tiered production system is very similar. So you start with a research seed lot, you progress to a master seed lot, from which you then create working seed lots from, and then eventually you create your production batch. This kind of a tiered system is applicable to GMP production. It's also applicable to magistral production or any GMP-like production scheme. Now, if we look at the upcoming and existing laws, regulations, and guidelines, the European Pharmacopoeia, section 5.31, states that the genetic stability of the phage is determined by sequencing a suitable number of purified harvests, followed by a comparison against the phage master seedlot sequence. If we look at the upcoming EMA draft guideline, which has been mentioned today a few times, they state that the full genome sequencing and genome annotation should be performed once for each established master seedlot. In case a working seedlot is established, the genome of the working seedlot should also be fully sequenced and compared to the master seedlot's nucleotide sequence, unless otherwise justified. And I will return to this last three words in a bit. Also, on the veterinary side, the EMA guidelines say for bacteriophages as well as matched bacterial hosts, the seedlot systems should ensure the genetic and phenotypic stability together with the viability of the material. So it's quite clear that not only phenotypic characterization of these phage products should be done, but also from the genomic point of view. But these guidelines still, especially the upcoming draft, they still leave a lot of room for interpretation. For example, when it's said that unless otherwise justified, how do we justify not sequencing an end product? And I will return to this in a moment. But just to look at the master seedlot from a genomic point of view. So it's the starting point for your phage production. And it establishes the reference genome for the production pipeline. And according to the guidelines, this master seedlot has to be well annotated and characterized. So you need to assemble it, annotate it, and analyze, and try to get as much of the hypothetical proteins into actual functional annotations. Now, if you look at the working seedlot, the idea behind the working seedlot is that it should be a clone of the master seedlot established from a single phage plague. And in the current guidelines, the idea reasonably is not to have this working seedlot completely fully assembled and analyzed in the same way that the master seedlot is. So the idea is that you would still sequence it if justified, and you would then compare your raw data against the master seedlot. And in bioinformatics, we call this comparison mapping. Now, the question still lingers that should we monitor these production batches or working seedlots? So I'm going to look a bit about how to control these mutations or any possible mutations and what risks there might be. So whenever you think about replication in a biological genetic entity, you will have the risk of mutations. And mutations can lead to unpredictable outcomes. You never know which part is being mutated. How is that reflected on the protein level, for example? And there's very sophisticated molecular machinery in prokaryotes and eukaryotes that try to minimize mutations during DNA replication. But these don't always work perfectly in prokaryotes or eukaryotes. And in phages in particular, we generally see higher mutation rates as compared to their host. So for example, if you look at lambda and its host E. coli, depending on the source, you get twice to 150 times higher mutation rates as compared to the host. And you always need to think about the fact that phages and bacteria are engaged in this billions of years old arms race, where phages try to overcome bacterial defenses and bacteria try to overcome the phage infections. So there's kind of incentive for phages to mutate and to keep variation in their population to survive these conditions. So there are several other factors that may increase these mutation rates. Well, I just mentioned host defenses, also external mutagens, the genome type, is it a double-stranded DNA phage or a single-stranded, for example. Co-infection, if you have a host with a prophage, which obviously you wouldn't want, but maybe sometimes you don't have a choice. Mobilizable elements in the bacterial host genome that might be incorporated into the phage genome, and simply the fact that you have so much unknown in phage genomes. A large proportion of phages are still called the phage dark matter, so we don't actually know what they do. So the bottom line here is that it's hard to control mutations, and when you do tiered production of phages, you introduce risks, because every time you create a new master seed lot, a working seed lot, or a production batch, you introduce a point where mutations can occur. So the phage DNA can and it will mutate over time. We can control it to an extent, and we should choose hosts that don't have prophages. We should choose hosts that have good proofreading polymerases and so on, but we can't control for everything, and we could just have, by chance, a mutation. And you always have to remember that with phages, if you have a mutation that gives the phage selective advantage from an evolutionary point of view, it can fixate in the population, unlike a non-living drug. So I want to ask the question that, are we prepared as a community to do genomic monitoring of phages? And looking at the current guidelines, it still seems a bit unclear. So on the high level, I think we should have community-wide discussions on what are the moments when sequencing these downstream lots is justifiable. So how can we justify not doing this sequencing? Also, the level and quality of the master seed lot characterization. Should we have a certain percentage of genes being annotated until we are happy with sufficiently having characterized these phages? Also, acceptable limits for variation in these working seed lots, synonymous mutations versus non-synonymous mutations, cutoffs for how big a proportion of reads is allowed to support a given mutation, and so on. On a more practical level, we need to prepare standardized bioinformatic workflows for these, and we need to be able to validate them, and we need to have quality control gates that are determined in these high-level requirements I just mentioned. And we need to have policies where authorized personnel are looking at these results and signing them, and creating documentation for keeping track of the mutations and quality. And we also need to have organizational policies that dictate the rules for the above. And in a GMP world, you would also need to have strict audit trails to track everything. So, brief look at the status quo. So how is genomic integrity currently being monitored? So in the worst case scenario, there is no genomic monitoring at all in phage production. In some cases, your typical monitoring might look something like this. So you would have a lab producing a batch, and this batch would be sent to a sequencing center, and then you would receive the raw data files. And these files would go through your bioinformatician or a bioinformatics department, who would then use a large number of different tools, most likely not tailored for this specific case, then creating lots of different documentation and results, which then need to be looked at by different personnel in your organization, and possibly have them signed through an external signature service. So what we've been working on at PrecisionPhage is our upcoming product called Phagenomics BatchQC. So the idea here is to bundle and democratize the whole genomic monitoring system into one neat package, which is easy to use. And the idea is not to replace your bioinformatician. You still need expertise in determining, for example, the thresholds and the cutoffs and the checkpoints for what is acceptable quality. And you also need to understand the results. But the idea is to give a central platform which all the personnel in your organization can actually use. and it's currently under development. So the idea is to enforce genomic integrity between batches and to spot contaminants. We have bioinformatic pipelines running behind the scenes, but they're abstracted behind a very easy to use graphical user interface. So you do not need to know how to code. You can customize it to your organization. So I mentioned before the working seed lots and the master seed lots and the production batches. You might have a different tiered system. So it's completely customizable to whatever tiered system you would like to use. You can also add metadata to your phages and customized quality control rules. We've also built an FDA CFR 21 Part 11 compliant signature system if you're doing GMP level production into the product. So you don't actually need to leave the product for doing signatures. We also have an exportable audit trail, a CSV file or an Excel file, which tracks all the actions done by your people in the organization. And as an end product of a batch, you can generate these certificates of analysis, which are PDFs, which can then be uploaded to your eQMS system, for example. We're compatible with PacBio, Illumina, and Nanopore technologies. And again, we offer very customizable organization-specific policies. And the idea is basically to streamline the whole genomic integrity monitoring system into one package. And especially if your organization scales up, this reduces risks that you might have from, for example, having a bottleneck in your bioinformatic analysis. So I'm going to finish with some outstanding questions that I would like to discuss as a community. So I'm very happy if we have people in the audience who would like to address these questions or talk about how you're currently doing phage production. So one central question is that should we establish standardized limits for variation, synonymous versus non-synonymous mutations, for example? Should there be minimum limits for the master seedlot characterization? How much should the replication host itself affect these decisions? So should we have specific requirements that what kind of a host is acceptable from a genomic point of view? And if we do establish some rules, should they be general or should they be phage family specific, for example? Because these phages, they differ a lot. It's much harder to annotate one phage family than another one. And finally, can we afford leaving out this genomic characterization? So what would be the actual justification for doing this? And with that, I would like to thank you for your company and happy to answer any questions.
Thank you very much, Ville, for your presentation. Indeed, a number of questions. Probably we won't have time to really discuss all of them, but if you would like to somehow react to the questions that Ville presented at the end, you are also, please feel free to reach out directly indeed to Ville. You can, Ville, leave your also contact details in the chat.
Sure.
We have a few, maybe even not questions, but maybe considerations or remarks. So I will just share them with you. Just a moment. It's a lot about mutations.
Mm-hmm.
I think that in general, we could say whether, you know, the question if mutations are good or bad in case of phages. I think, yeah, the remark, the first remark was that mutations is what makes phage therapy better than chemical antimicrobials. They are not a problem, they are the goal. And I think also the remark number, with number 3 is also similar, that it would be damaging to phage therapy to deny phage mutations. Instead, we need to push regulators the fact that it is normal and necessary. Phages mutate so much more in vivo after applications. Do you want to address it somehow?
Sure. I definitely agree that mutations are a great advantage of phages, especially when applied to a patient. But I would still argue that you need to know what you are applying to the patient before you do so. So if you use your production bacterial host and that host for example, happens to have an antibiotic resistance gene, which has gone unnoticed somehow. And that gene is incorporated into the phage genome, which does happen. It's unlikely, but it can happen. And the fact that you might then have selective advantage for this kind of a phage in the patient, I would argue that you would need definitely to know what is your end product, even if it would mutate then at the patient level. That's kind of a different matter. But I think we all want to know what the end product actually is. Otherwise, we are flying blind, I would say.
Yes. Point or question number 4. Yeah, I think it relates also to a bit to number 3. It's also again about mutations. Do you want to address it somehow? The comment in number 4.
So cell-free doesn't stop mutations, replication errors still happen. That's definitely true. Limits number of phages you can produce. I'm not completely sure I understand the question.
Yeah, I know that writing questions, it's not always the best solution, but at least it's easier for us to manage a bit the time. There is a question number 2. What about cell-free phage production to avoid unintentional mutations?
Yeah, that's an interesting point. Mutations, of course, polymerases still make mistake even if you do cell-free replication, in vitro replication. Sure, but yeah. So I would still argue that it is important to monitor what you produce, even if you do it cell-free.
Okay. Okay, I think I don't see any additional question. It seems to me there is like some discussion ongoing. Indeed, there are some replies also to the questions about mutations in the chat. Yeah, so thank you very much, Ville, once again. If there are any further questions, please put them in the chat. We still have 15 minutes, so Ville will reply for sure in the chat. And